Chapter 2 Sexual Reproduction Flowering Plants 1. Dash are morphological and embryological marvels. Flowers, flowers, flowers. 2. Dash and dash changes initiate the development of the floral primordium. Hormonal and structural, hormonal and structural, hormonal and structural. 3. Inflorescences are formed after the appearance of the flower. False, false, false. 4. A three-dimensional cut section of an anther is shown in diagram. I. Identify its parts A, B, C, and D. Diggle. 1. A. Pollen grains, B. Pollen sacs, C. Line of dehiscence, D. Filament. Diggle. 1. A. Pollen grains, B. Pollen sacs, C. Line of dehiscence, D. Filament. Diggle. 1. A. Pollen grains, B. Pollen sacs, C. Line of dehiscence, D. Filament. 5. Parts of the stamen are filament and anther. Filament and anther. Filament and anther. 6. The proximal, distal end of the filament is attached to dash or dash of the flower. Proximal, thalamus or petal. Proximal, thalamus or petal. Proximal, thalamus or petal. 7. A typical angiosperm anther is anilobed, bilobed and didicous, tetrathecous condition. Bilobed, didicous, bilobed, didicous, bilobed, didicous. 8. A longitudinal, vertical groove runs lengthwise separating the theca. Longitudinal, longitudinal, longitudinal. 9. Anther consists of how many microsporangia? 4, 4, 4, 10. Each lobe contains how many microsporangia? 2, 2, 2, 11. Microsporangia develop further and become dash. Pollen sacs, pollen sacs, pollen sacs. 12. A transverse section of a young anther is shown in diagram. 2. Label A to F. Diagram. 2. A. Connective, B. Epidermis, C. Endothecium, D. Sporogenous tissue, A. Tapetum, F. Middle layers. Diagram. 2. A. Connective, B. Epidermis, C. Endothecium, D. Sporogenous tissue, A. Tapetum, F. Middle layers. Diagram. 2. A. Connective, B. Epidermis, C. Endothecium, D. Sporogenous tissue, A. Tapetum, F. Middle layers. 13. Enlarged view of one microsporangium is shown in diagram. 3. Identify its wall layers A to A. Diagram. 3. A. Epidermis, B. Endothecium, C. Middle layers, D. Microspore mother cells, A. Tapetum. Diagram. 3. A. Epidermis, B. Endothecium, C. Middle layers, D. Microspore mother cells, A. Tapetum. Diagram. 3. A. Epidermis, B. Endothecium, C. Middle layers, D. Microspore mother cells, A. Tapetum. 14. Wall layers of microsporangium are epidermis, endothecium, middle layer, and tapetum. Epidermis, endothecium, middle layer, and tapetum. Epidermis, endothecium, middle layer, and tapetum. 15. A typical microsporangium is oval in outline. False, false, false. 16. Dash nourishes the developing pollen grain. Tapetum, tapetum, tapetum. 17. The outer three layers perform the function of protection and help in dehiscence of the anther. Protection and help in dehiscence of the anther. Protection and help in dehiscence of the anther. 18. Tapetum has light, dense cytoplasm and more than one nucleus and polyploidy. Dense, more than one. Dense, more than one. Dense, more than one. 19. Tapetum function. Nourish developing pollen. Nourish developing pollen. Nourish developing pollen. 20. Middle layer function. They are generally ephemeral and are crushed during meiosis in the microspore mother cell. They are generally ephemeral and are crushed during meiosis in the microspore mother cell. They are generally ephemeral and are crushed during meiosis in the microspore mother cell. 21. When the anther is young, a group of compactly arranged cells called dash occupies the center of each microsporangium. Sporogenous tissue. Sporogenous tissue. Sporogenous tissue. 22. Only a few cells of the sporogenous tissue are capable of giving rise to a microspore tetrad. T.F. False. 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 23. MMC is also called dash. Megasporocyte. 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 24. The two anther lobes are attached to each other by dash. Connective, given in diagram. Connective, given in diagram. Connective, given in diagram. 25. Dash help in dehiscence of the anther due to their hygroscopic nature. Endothecium. 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 26. Tapetum surrounds the sporogenous tissue. True, given in diagram. True, given in diagram. True, given in diagram. True, given in diagram. 27. What is microsporogenesis? The process of formation of microspores from a pollen mother cell. The process of formation of microspores from a pollen mother cell. The process of formation of microspores from a pollen mother cell. 28. As the anthers mature and hydrate, the microspores dissociate from each other and develop into pollen grains. False, dehydrate. False, dehydrate. False, dehydrate. 29. A few stages of a microspore maturing into a pollen grain are shown in diagram. 4. Identify A, B, and C. Diagram. 4. A. Asymmetric spindle. B. Vegetative cell. C. Generative cell. Diagram. 4. A. Asymmetric spindle. B. Vegetative cell. C. Generative cell. Diagram. 4. A. Asymmetric spindle. B. Vegetative cell. C. Generative cell. 30. Pollen grains are released with the dash of the anther. Dehiscence. 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 31. Pollen grains represent the male gametophytes, male sporophyte, male gametophytes. Male gametophytes. Male gametophytes. 32. Pollen grain measures about -mm in diameter. 0.025-0.050. 0.025-0.050. 0.025-0.050. 33. Its wall is single-layered. False, double-layered. False, double-layered. False, double-layered. 34. The hard outer layer called dash is made up of dash. Exine, sporophyllin. Exine, sporophyllin. Exine, sporophyllin. 35. Dash is one of the most resistant organic materials known. Sporophyllin. 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 36. No enzyme that degrades sporophyllin is so far known. True. 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 37. The part of exine where sporophyllin is absent is germ pores. Germ pores. Germ pores. 38. Pollen grains are well preserved as fossils due to presence of sporophyllin. Presence of sporophyllin. Presence of sporophyllin. 39. Exine, intine exhibit a fascinating array of pattern and design. Exine. 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 40. Intine is a thin, thick layer made up of dash and dash. Thin, cellulose and pectin. Thin, cellulose and pectin. Thin, cellulose and pectin. 41. Mature pollen grain contains two cells, namely vegetative cell and generative cell. Vegetative cell and generative cell. Vegetative cell and generative cell. 42. Vegetative, generative cell is bigger. Vegetative. 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 43. Irregular shaped nucleus is present in vegetative, generative cell. Vegetative cell. Vegetative cell. Vegetative cell. 44. Vegetative cell have abundant food reserve. True. 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 45. Generative cell is dash shaped. Spindle. 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 46. Generative, vegetative cell floats in the cytoplasm of generative, vegetative cell. Generative cell, vegetative. Generative cell, vegetative. Generative cell, vegetative. 47. Generative cell have dense cytoplasm and nucleus. True. 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 48. In less than -% of angiosperms, pollen grains are shed at 3-cell stage. 40. 40. 40. 49. Dash cell divide to give 3-cell stage. Generative. 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 50. Pollen grains may cause asthma and bronchitis. True. 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 51. Carrot grass, also called dash, came into India as a contaminant with imported dash. False. 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 52. Parthenium do not cause pollen allergy. True. 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 53. Pollen consumption has been claimed to increase the performance of dash and dash. Athletes and racehorses. Athletes and racehorses. Athletes and racehorses. 54. Pollen grains have to land on the dash before they lose viability if they have to bring about fertilization. Stigma, stigma, stigma. 55. Pollen viability depends on prevailing temperature and humidity. True. 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 56. In rice and wheat, pollen grains lose viability within _ minutes. 30. 30. 30. 57. Families whose pollen remains viable for months are Rosaceae, Leguminosae and Solanaceae. Rosaceae, Leguminosae and Solanaceae. Rosaceae, Leguminosae and Solanaceae. 58. Pollen grains are stored in dash for years. Liquid nitrogen, liquid nitrogen, liquid nitrogen. 59. Liquid nitrogen temperature is -196 degrees Celsius. -196 degrees Celsius. minus 196 degrees Celsius. 60. Pollen grains are preserved for years by using the technique of dash. Cryopreservation. 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 61. Pollen banks cannot be used in crop breeding programs. False. 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 62. A dissected flower of hibiscus is shown in diagram. 5. Identify its parts A, B, C and D. Diagram. 5. A. Stigma. B. Style. C. Ovary. D. Thalamus. Diagram. 5. A. Stigma. B. Style. C. Ovary. D. Thalamus. Diagram. 5. A. Stigma. B. Style. C. Ovary. D. Thalamus. 63. A multicapillary, syncapas pistil of a flower is shown in diagram. 6. Identify the species and label A and B. Diagram. 6. Papava, A. Stigma. B. Syncapus ovary. Diagram. 6. Papava, A. Stigma. B. Syncapus ovary. Diagram. 6. Papava, A. Stigma. B. Syncapus ovary. 64. A multicapillary apocarpus gynoecium of a flower is shown in diagram. 7. Identify the species and label A. Diagram. 7. Michelia, A. carpels. Diagram. 7. Michelia, A. carpels. Diagram. 7. Michelia, A. carpels. 65. Monocapillary condition means single pistil, single pistil, single pistil. 66. Pistils may be fused together called dash or free called dash, syncarpus, apocarpus, syncarpus, apocarpus, syncarpus, apocarpus. 67. The basal bulged part of pistil is ovary, ovary, ovary. 68. Ovarian cavity is also called locule, locule, locule. 69. Placenta is located outside the ovarian cavity. T. False, false, false. 70. Megasporangia are commonly called dash. Ovules, ovules, ovules. 71. Megasporangia arise from placenta. True, true, true. 72. Apocarpus XI. Michelia, Michelia, Michelia. 73. Syncarpus XI. Papava, Papava, Papava. 74. Papava and Michelia are monocapillary. False, false, false. 75. Single ovule is present in wheat, paddy and mango. Wheat, paddy and mango. Wheat, paddy and mango. 76. Multiple ovules are present in papaya, watermelon and orchids. Papaya, watermelon and orchids. Papaya, watermelon and orchids. 77. Most common type of ovule is anitrous ovule, anitrous ovule, anitrous ovule. 78. Type of division in which karyokinesis, nuclear division, in a cell is not followed by immediate cytokinesis results in syncytium, syncytium, syncytium. 79. Anatropous ovule meaning in which curvature during development results in displacement of the micropyle to a position adjacent to the funiculus base, in which curvature during development results in displacement of the micropyle to a position adjacent to the funiculus base, in which curvature during development results in displacement of the micropyle to a position adjacent to the funiculus base. 80. A diagrammatic view of an anatropous ovule is shown in diagram. 8. Label A to L. Diagram. 8. A. Hilum. B. Funicle. C. Micropyle. D. Micropylar pole. A. Outer integument. F. Inner tertiary. G. Nucleus. H. Embryo sac. I. Chalazal pole. Diagram. 8. A. Hilum. B. Funicle. C. Micropyle. D. Micropylar pole. A. Outer integument. F. Inner tertiary. G. Nucleus. H. Embryo sac. I. Chalazal pole. Diagram. 8. A. Hilum. B. Funicle. C. Micropyle. D. Micropylar pole. A. Outer integument. F. Inner tertiary. G. Nucleus. H. Embryo sac. I. Chalazal pole. 81. Ovule is attached to placenta by dash. Funicle. 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 82. The body of the ovule fuses with funicle in the region called dash. Hilum. 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 83. Ovule has how many integuments? 1 or 2. 1 or 2. 1 or 2. 84. Function of integuments. Protection. 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 85. Integuments encircle the nucellus except at dash. Micropyle. 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 86. Opposite to micropyle is the dash end. Chalazal. 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 87. Micropyle represents the basal part of the ovule. False. 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 88. Dash is enclosed within the integuments. Nucellus. 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 89. A diagrammatic representation of a mature embryo sac is shown in diagram. 9. Identify X and Y ends of the ovule. Label A to F. Diagram. 9. X, chalazal end, Y, micropylar end, A, antipodals, B, polar nuclei, C, central cell, D, egg. A, synegids, F, filiform apparatus. Diagram. 9. X, chalazal end, Y, micropylar end, A, antipodals, B, polar nuclei, C, central cell, D, egg. A, synegids, F, filiform apparatus. Diagram. 9. X, chalazal end, Y, micropylar end, A, antipodals, B, polar nuclei, C, central cell, D, egg. A, synegids, F, filiform apparatus. 90. Female gametophyte is also called dash. Embryo sac. Embryo sac. Embryo sac. 91. Cells of the nucellus have abundant reserve food materials. True. 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 92. An ovule generally has a single embryo sac. True. 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 93. Ovules generally differentiate a single MMC from the chalazal, micropylar region of nucellus. Micropylar. 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 94. MMC have thin, dense cytoplasm and inconspicuous, prominent nucleus. Dense, prominent. Dense, prominent. Dense, prominent. 95. Meiosis results in production of how many megaspores? 4. 4. 4. 96. Out of 4, how many megaspore are functional? 1. 1. 1. 97. What is monosporic development? Embryo sac form from single megaspore rest all degenerate. Embryo sac form from single megaspore rest all degenerate. Embryo sac form from single megaspore rest all degenerate. 98. The nuclear divisions in the function megaspore are free nuclear. True. 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 99. What does free nuclear division mean? Type of division in which karyokinesis is not followed by immediate cytokinesis. Type of division in which karyokinesis is not followed by immediate cytokinesis. Type of division in which karyokinesis is not followed by immediate cytokinesis. 100. Egg apparatus consists of 2 synegids in a egg cell. 2 synegids in a egg cell. 2 synegids in a egg cell. 101. Egg apparatus is present at micropylar, chalazal end. Micropylar, micropylar, micropylar. 102. Synegids have special cellular thickenings at the micropylar tip called filiform apparatus. Filiform apparatus. Filiform apparatus. 103. Dash no of cells are present in chalazal end and are called dash. 3. Antipodal cells. 3. Antipodal cells. 3. Antipodal cells. 104. Function of filiform apparatus. Guide the pollen tube into the synegids. Guide the pollen tube into the synegids. Guide the pollen tube into the synegids. 105. A typical angiosperm embryo sac at maturity shows 7 nucleated, 8 self-condition. False. 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 106. Pollen grains are non-motile. True. 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 107. Depending on source of pollen, the 3 types of pollination are autogamy, geitonogamy, xenogamy. Autogamy, geitonogamy, xenogamy. Autogamy, geitonogamy, xenogamy. 108. Diagram. 10 represents a plant with 2 types of flowers. Identify the types of flowers A and B. A. Chasmogamous flower. B. Cleistogamous flower. A. Chasmogamous flower. B. Cleistogamous flower. A. Chasmogamous flower. B. Cleistogamous flower. 109. X of autogamy are Viola, Oxalis and Camelina. Viola, Oxalis and Camelina. Viola, Oxalis and Camelina. 110. In a flower which opens and exposes anthers and stigma, complete autogamy is almost always present. False. 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 111. Autogamy requires synchrony between dash and dash, pollen release and stigma receptivity, pollen release and stigma receptivity, pollen release and stigma receptivity. 112. In chasmogamous flowers, anthers and stigma lie close to each other. False. 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 113. Cleistogamous, chasmogamous flowers are invariably autogamous. Cleistogamous. 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 114. Cleistogamy is disadvantageous as there is no chance for cross-pollination. True. 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 115. Dash flowers produce a SHOUD seed set even in the absence of pollinators. Cleistogamous, autogamous. Cleistogamous, autogamous. Cleistogamous, autogamous. 116. What is geitonogamy? Transfer of pollen grains from the anther to the stigma of another flower of the same plant. Transfer of pollen grains from the anther to the stigma of another flower of the same plant. Transfer of pollen grains from the anther to the stigma of another flower of the same plant. 117. Gytonogamy is functionally autogamy, cross-pollination. 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 118. Gytonogamy is genetically similar to autogamy, cross-pollination. Autogamy, autogamy, autogamy. 119. Only type of pollination which brings genetically diff types of pollen grains to the stigma is xenogamy. 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 120. Two abiotic and one biotic agents for pollination are abiotic, water and wind, biotic, animals. Abiotic, water and wind, biotic, animals. Abiotic, water and wind, biotic, animals. 121. Majority of plants use abiotic, biotic agents for pollination. Biotic. 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 122. Pollination by dash is more common amongst abiotic pollinations. Wind. 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 123. Wind pollinators produce sticky pollen grains so that they stick to stigma. False. 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 124. Pollen grain of wind pollinators is light. True. 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 125. Feathery stigma and well-exposed stamens are features of wind pollinators. Wind pollinators. Wind pollinators. 126. Wind pollinators have how many ovules in the ovary? 1. 1. 1. 127. Packed inflorescence is not a feature of wind pollinators. False. 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 128. Dash of corn cob are stigma and style which wave in the wind. Tassels. 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 129. X of wind pollination. Grasses and maize, corn. Grasses and maize, corn. Grasses and maize, corn. 130. Pollination by water is limited to dash no of genera, mostly monocot, dicot. 30. Monocot. 30. Monocot. 30. Monocot. 131. Pollination by water in a plant is shown in diagram. Eel. Identify the plant species in label A, B, C and D. Diagram. Eel, Vallisneria, A, female flower, B, stigma, C, male flower, D, female flower. Diagram. Eel, Vallisneria, A, female flower, B, stigma, C, male flower, D, female flower. Diagram. Eel, Vallisneria, A, female flower, B, stigma, C, male flower, D, female flower. 132. Distribution of bryophytes, algae and pteridophytes is limited because need of water for fertilization, need of water for fertilization, need of water for fertilization. 133. X of freshwater pollinated plants. Vallisneria and hydrilla. Vallisneria and hydrilla. Vallisneria and hydrilla. 134. X of marine water pollinated plants. Seagrasses. 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 135. Seagrass X. Zostera. 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 136. Aquatic plants not pollinated by water are water hyacinth and water lily. Water hyacinth and water lily. Water hyacinth and water lily. 137. In dash female flower reach to the surface of water. Vallisneria. 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 138. In dash female flower are submerged into water. Seagrasses. 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 139. In dash epihydrophyly is present. Vallisneria. 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 140. Hypohydrophyly is present in Zostera. 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 141. Pollen grains of seagrasses are long, short and dash-like, long, ribbon-like, long, ribbon-like, long, ribbon-like. 142. Pollen grains in seagrasses are carried actively. False. 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 143. In most of the water-pollinated species, pollen grains are protected from wetting by dash. Mucilaginous covering. Mucilaginous covering. Mucilaginous covering. 144. Flowers are brightly colored in wind and water-pollinated plants. False. 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 145. Common biotic pollinating agents are bees, butterflies, flies, beetles, wasps, moths, ants, sunbirds, hummingbirds, bats. Bees, butterflies, flies, beetles, wasps, moths, ants, sunbirds, hummingbirds, bats. Bees, butterflies, flies, beetles, wasps, moths, ants, sunbirds, hummingbirds, bats. 146. Dash are the dominant biotic pollinating agents. Bees, bees, bees. 147. X of lizards reported as pollinating agents is gecko lizard and garden lizard. Gecko lizard and garden lizard. Gecko lizard and garden lizard. 148. Primate reported as pollinating agent is lemur. 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 149. Arboreal rodents can't act as pollinating agents. False. 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 150. Flowers of animal pollinating plants are not specifically adapted for particular species. False. 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 151. Majority of insect pollinated flowers are small. False. 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 152. What happens when insect pollinated flowers are small? They cluster into an inflorescence, they cluster into an inflorescence, they cluster into an inflorescence. 153. Animals are attracted to flowers by dash and dash, color and fragrance, color and fragrance, color and fragrance. 154. Flowers produce foul odors to attract dash and dash. Flies and beetles. Flies and beetles. Flies and beetles. 155. Tallest flower is of dash feet and its name is 6 feet, Amorphophallus. 6 feet, Amorphophallus. 6 feet, Amorphophallus. 156. Largest inflorescence and largest flower are Amorphophallus and Rafflesia. Amorphophallus and Rafflesia. Amorphophallus and Rafflesia. 157. In Amorphophallus, floral rewards given by plant is safe place to lay eggs. Safe place to lay eggs. Safe place to lay eggs. 158. Yucca and dash cannot complete their life cycle with each other. Moth name Pronuba. Moth name Pronuba. Moth name Pronuba. 159. Moth deposits its egg in dash of the ovary. Locule. 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 160. Define nectar robbers. Many insects may consume pollen or nectar without bringing about pollination. Such floral visitors are called pollen nectar robbers. Many insects may consume pollen or nectar without bringing about pollination. Such floral visitors are called pollen nectar robbers. Many insects may consume pollen or nectar without bringing about pollination. Such floral visitors are called pollen nectar robbers. 161. Majority of flowering plants are unisexual. False. 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 162. Continued self-pollination result in dash. Inbreeding depression. Inbreeding depression. Inbreeding depression. 163. 4 outbreeding devices are I. Unisexuality of flower. E. Dichogamy different maturation time of Andrecium and Gynoecium. E. Self-incompatibility inhibition of pollen germination of the same plant. IV. Anther and stigma are at different positions so that pollen cannot come in contact. I. Unisexuality of flower. E. Dichogamy different maturation time of Andrecium and Gynoecium. E. Self-incompatibility inhibition of pollen germination of the same plant. IV. Anther and stigma are at different positions so that pollen cannot come in contact. I. Unisexuality of flower. E. Dichogamy different maturation time of Andrecium and Gynoecium. E. Self-incompatibility inhibition of pollen germination of the same plant. IV. Anther and stigma are at different positions so that pollen cannot come in contact. 164. Unisexuality prevent dash but not dash. Autogamy, gytnogamy, autogamy, gytnogamy, autogamy, gytnogamy. 165. Papaya is monoecious, dioecious. 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 166. Monoecious X. Castor and maize. Castor and maize. Castor and maize. 167. Pollination guarantees the transfer of the right type of pollen grains. False. 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 168. Pistil cannot recognize whether pollen is of the right type or not. False. 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 169. In two-celled pollen, the generative cell divides during dash. Growth of pollen tube in the stigma. Growth of pollen tube in the stigma. Growth of pollen tube in the stigma. 170. Pollen tube enters ovule through chalazal, micropyla end. Micropyla, micropyla, micropyla. 171. Dash guides the entry of the pollen tube. Filiform apparatus. Filiform apparatus. Filiform apparatus. 172. To study pollen germination, a drop of -% solution of is taken on a pollen-dusted glass slide. 10. Sugar. 10. Sugar. 10. Sugar. 173. Pollen grain of Crotalaria cannot be studied on a glass slide. False. 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 174. After waiting for dashmin, pollen grains are studied under high, low-power microscope. 15 to 30, low. 15 to 30, low. 15 to 30, low. 175. What is emasculation? Removal of anther from floral bud before anther dehiscence. Removal of anther from floral bud before anther dehiscence. Removal of anther from floral bud before anther dehiscence. 176. Emasculated flowers are covered with a bag made of butter paper, bagging. Butter paper, bagging. Butter paper, bagging. 177. There is no need for emasculation in dash flowers. Unisexual female. Unisexual female. Unisexual female. 178. Stages in embryo development in a dicot plant are shown in diagram. 12. Label A to F. Diagram. 12A. Globular embryo. B. Heart-shaped embryo. C. Suspensor. D. Radical. E. Cotyledon. F. Plumule. Diagram. 12A. Globular embryo. B. Heart-shaped embryo. C. Suspensor. D. Radical. E. Cotyledon. F. Plumule. Diagram. 12A. Globular embryo. B. Heart-shaped embryo. C. Suspensor. D. Radical. E. Cotyledon. F. Plumule. 179. Triple fusion happens in double fertilization. True. 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 180. Pollen tube releases two male gametes into the cytoplasm of the synegid. True. 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 181. The male gametes fuse with dash and dash, egg and central cell, egg and central cell, egg and central cell. 182. Dash is an event unique to angiosperms. Double fertilization. Double fertilization. Double fertilization. 183. Central cell after fusion becomes PEC primary endosperm cell. PEC primary endosperm cell. PEC primary endosperm cell. 184. Suspensor grows to push the embryo away from the chalazal, micropylar end. Micropylar. 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 185. Suspensor helps to transfer dash to the embryo. Nutrients. 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 186. Embryo development precedes endosperm development. False. 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 187. Most common type of endosperm development is free nuclear, free nuclear, free nuclear. 188. Endosperm is used for dash the developing embryo. Nourishing, nourishing, nourishing. 189. Free nuclear endosperm X. Coconut water. Coconut water. Coconut water. 190. Cellular endosperm is formed before free nuclear endosperm. False. 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 191. Endosperm is completely consumed in P. Groundnut beans. P. Groundnut beans. P. Groundnut beans. 192. Endosperm persists in mature seeds in castor and coconut. Castor and coconut. Castor and coconut. 193. Endosperm persists in cereals. True. 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 194. A typical dicot embryo is shown in diagram. 13. Label A to A. Diagram. 13A. Plumule. B. Cotyledons. C. Hypocotyl. D. Radical. A. Root cap. Diagram. 13A. Plumule. B. Cotyledons. C. Hypocotyl. D. Radical. A. Root cap. Diagram. 13A. Plumule. B. Cotyledons. C. Hypocotyl. D. Radical. A. Root cap. 195. An LS of an embryo of grass is shown in diagram. 14. Label A to G. Diagram. 14A. Scutellum. B. Coleoptile. C. Shoot apex. D. Epiblast. A. Radical. F. Root cap. G. Coleorizer. Diagram. 14A. Scutellum. B. Coleoptile. C. Shoot apex. D. Epiblast. A. Radical. F. Root cap. G. Coleorizer. Diagram. 14A. Scutellum. B. Coleoptile. C. Shoot apex. D. Epiblast. A. Radical. F. Root cap. G. Coleorizer. 196. Embryo develop at the micropylar, chalazal end of the embryo sac. Micropylar, micropylar, micropylar. 197. Zygote division is independent of the amount of endosperm present. False. 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 198. The early stage of embryogeny is different in both monocots and dicots. False. 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 199. The stages of embryogeny from zygote to mature embryo are zygote to proembryo to globular to heart-shaped to mature embryo. Zygote to proembryo to globular to heart-shaped to mature embryo. Zygote to proembryo to globular to heart-shaped to mature embryo. 200. Name the two parts of embryo. Embryonal axis and cotyledons. Embryonal axis and cotyledons. Embryonal axis and cotyledons. 201. Embryonal axis above the level of attachment of cotyledon is dash and below is dash. Epicotyl, hypocotyl, epicotyl, hypocotyl, epicotyl, hypocotyl. 202. Epicotyl terminates with dash or dash plumule or shoot tip, plumule or shoot tip, plumule or shoot tip. 203. Hypocotyl is cylindrical. True, true, true. 204. Hypocotyl terminates at its lower end in dash or dash, radical, root tip, radical, root tip, radical, root tip. 205. Radical is covered by root cap, root cap, root cap. 206. In grass family, cotyledon is called dash, scutellum, scutellum, scutellum. 207. Scutellum is situated on then dash side of the embryonal axis. Lateral, lateral, lateral. 208. In monocot, root cap is enclosed in dash. Clearizer, clearizer, clearizer. 209. Clearizer is a differentiated sheath. False, false, false. 210. Epicotyl and a few leaf primordial are enclosed inside dash, coleoptile, coleoptile, coleoptile. 211. Coleoptile is a hollow, solid dash structure. Hollow, folair, hollow, folair, hollow, folair. 212. Dash is the final product of sexual reproduction. Seed, seed, seed. 213. Fertilized ovule mean. Seed, seed, seed. 214. Typical seed contains dash, dash, and dash, seed coat, cotyledons, embryonal axis, seed coat, cotyledons, embryonal axis, seed coat, cotyledons, embryonal axis. 215. Structure of some seeds is shown in diagram. 15. Label A to F and P to V. Diagram. 15A, seed coat, B, cotyledon, C, shoot apical meristem, D, hypocotyl root axis, A, root tip. F, endosperm, P, pericarp. Q, endosperm, R, scutellum, S, coleoptile, T, plumule U, radical V, clearizer. Diagram. 15A, seed coat, B, cotyledon, C, shoot apical meristem, D, hypocotyl root axis, A, root tip. F, endosperm, P, pericarp. Q, endosperm, R, scutellum, S, coleoptile, T, plumule U, radical V, clearizer. Diagram. 15A, seed coat, B, cotyledon, C, shoot apical meristem, D, hypocotyl root axis, A, root tip. F, endosperm, P, pericarp. Q, endosperm, R, scutellum, S, coleoptile, T, plumule U, radical V, clearizer. 216. Non-albuminous seed doesn't have endosperm, endosperm, endosperm. 217. Residual, persistent nucellus is called perisperm, perisperm, perisperm. 218. Non-albuminous seed X, P, groundnut, P, groundnut, P, groundnut. 219. Albuminous seeds X, wheat, maize, barley, castor. Wheat, maize, barley, castor. Wheat, maize, barley, castor. 220. Perispermic seeds X, black pepper and beet, black pepper and beet, black pepper and beet. 221. X albuminous means a seed that contains albumin. False, false, false. 222. Integuments of ovules develop into dash. Seed coat, seed coat, seed coat. 223. Micropyle remains as a small pore in the seed coat. True, true, true. 224. As the seeds mature, the seed becomes dry, moist and they contain dash % of moisture by mass. Dry, 10 to 15%, dry, 10 to 15%, dry, 10 to 15%. 225. Dash facilitates the entry of water and oxygen for germination. Micropyle, micropyle, micropyle. 226. The transformation of ovules into seeds and ovary into fruit proceeds simultaneously. True, true, true. 227. Wall of the fruit is called pericarp, pericarp, pericarp. 228. Mustard fruit is fleshy, dry, 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 dry. 229. Groundnut fruit is fleshy, dry, 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 dry. 230. Is there any relationship between the number of ovules in an ovary and the number of seeds present in a fruit? Number of seeds present in the fruit will be equal to the number of ovules present in the ovary. Number of seeds present in the fruit will be equal to the number of ovules present in the ovary. Number of seeds present in the fruit will be equal to the number of ovules present in the ovary. 231. Define false fruits. Fruit developed from part other than ovary. Fruit developed from part other than ovary. Fruit developed from part other than ovary. 232. X of fruits which developed from thalamus are apple, strawberry and cashew. Apple, strawberry and cashew. Apple, strawberry and cashew. 233. False fruits of apple and strawberry are shown in diagram. 16. Label A, B, C, D and P. Q. Diagram. 16A, thalamus, B, seed, C, endocarp, D, mesocarp, P, thalamus, Q, achene. Diagram. 16A, thalamus, B, seed, C, endocarp, D, mesocarp, P, thalamus, Q, achene. Diagram. 16A, thalamus, B, seed, C, endocarp, D, mesocarp, P, thalamus, Q, achene. 234. Parthenocarpic fruit X. Banana, banana, banana. 235. Dash is the basis of our agriculture. Seed, seed, seed. 236. Dash and dash of mature seeds are crucial for the storage of seeds, which can be used as food throughout the year and also to raise a crop in the next season. Dehydration and dormancy. Dehydration and dormancy. Dehydration and dormancy. 237. Seeds of a large number of species lose viability in a few months. False. 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 238. Only some seeds can remain alive for hundreds of years. True. 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 239. The oldest viable seed is of excavated from dash. Lupinus arcticus, Arctic tundra. Lupinus arcticus, Arctic tundra. Lupinus arcticus, Arctic tundra. 240. The seed of lupine germinated after dash years of dormancy. 10,000. 10,000. 10,000. 241. Seed of a date palm, namely dash had a viability of dash years and was discovered at dash near dash. Phoenix dactylifera, 2000, King Herod's Palace, Dead Sea. Phoenix dactylifera, 2000, King Herod's Palace, Dead Sea. Phoenix dactylifera, 2000, King Herod's Palace, Dead Sea. 242. Plant having fruits with large no of tiny seeds X are orchids, Orobanche, striga, ficus. Orchids, Orobanche, striga, ficus. Orchids, Orobanche, striga, ficus. 243. Dash and dash are parasitic plant species in which each fruit contains thousands of tiny seeds. Orobanche and striga. Orobanche and striga. Orobanche and striga. 244. What is apomixis? Production of seeds without fertilization. Production of seeds without fertilization. Production of seeds without fertilization. 245. Apomixis is present in Asteraceae in grasses. Asteraceae in grasses. Asteraceae in grasses. 246. Apomixis is a form of asexual, sexual reproduction that mimic asexual, sexual reproduction. Asexual, sexual, asexual, sexual, asexual, sexual. 247. Diploid egg formation without reduction, division and development into an embryo can be called apomixis. True. 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 248. One ovule can contain many embryos. True. 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 249. What is polyembryony? Occurrence of more than one embryo in a seed. Occurrence of more than one embryo in a seed. Occurrence of more than one embryo in a seed. 250. Nachella polyembryony X. Citrus and mango. Citrus and mango. Citrus and mango. 251. The hybrid characters do not segregate in the progeny plants. False. 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 252. Apomictic seeds can reduce the cost of production for farmers. True. 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 253. Why seeds of orchids symbiotically associate with mycorrhiza? Seeds of orchids are tiny and have no food reserves. This is why they have to bond symbiotically with fungi to form mycorrhiza for germination. Seeds of orchids are tiny and have no food reserves. This is why they have to bond symbiotically with fungi to form mycorrhiza for germination. Seeds of orchids are tiny and have no food reserves. This is why they have to bond symbiotically with fungi to form mycorrhiza for germination. 254. Why are coconut trees found near sea beaches? Coconuts are best dispersed through seawater. Reasons: I. Coconut has mesocarp made of husk which helps to float. E. Coconut have large food reserve material which helps for survival for long time. Coconuts are best dispersed through seawater. Reasons: I. Coconut has mesocarp made of husk which helps to float. E. Coconut have large food reserve material which helps for survival for long time. Coconuts are best dispersed through seawater. Reasons: I. Coconut has mesocarp made of husk which helps to float. E. Coconut have large food reserve material which helps for survival for long time. 255. Ploidy level of synergids, zygote, and pen respectively are N, 2N, and 3N. N, 2n, and 3n, n, 2n, and 3n. 256. Large colorful, fragrant flowers with nectar are seen in insect, bird-pollinated flowers. Insect, insect, insect. 257. Examples of organisms having isogamids are algae, Cladophora, Chlamydomonas and Eulothrix fungi, Rhizopus and Syncytrium. Algae, Cladophora, Chlamydomonas and Eulothrix fungi, Rhizopus and Syncytrium. Algae, Cladophora, Chlamydomonas and Eulothrix fungi, Rhizopus and Syncytrium. 258. Examples of organisms having heterogamids. Fucus, Volvox, Chara, all bryophytes, pteridophytes, gymnosperms, and angiosperms. Fucus, Volvox, Chara, all bryophytes, pteridophytes, gymnosperms, and angiosperms. Fucus, Volvox, Chara, all bryophytes, pteridophytes, gymnosperms, and angiosperms. 259. Each PMC gives rise to 4 pollen grains. True, true, true. 260. Examples of plants having both male and female flowers on the same plant. Castor and maize, castor and maize, castor and maize.